Go.
Go.
Yeah.
Hello, I'm MGMauk from Philadelphia, and I'm presenting my review and classification of paper number 10 in Group 1 entitled Continued Evolution of West Nile Virus, Houston, Texas, USA, 2002 to 2012 by B.R. Mann at the University of Texas Medical Branch, Galveston, and published in Emerging Infectious Diseases, September 2013. First, a little perspective on the significance of the work. West Nile virus was introduced into the U.S. in 1999. It rapidly spread to almost all parts of the country. Surveillance studies such as this provide data on the evolution, transmission dynamics, and ecology of West Nile virus. Since 1999, it is estimated that in the U.S. there have been over 3 million human infections, including 1,500 associated fatalities. The virus' genetic and antigenic heterogeneity also figures in vaccine and therapy development. This was obviously not a typical clinical study, so it didn't really lend itself to the classification flowchart we've seen. Nevertheless, it does share many features common to the types of studies covered in class, which I'll now briefly discuss. I would classify the work as descriptive and observational. There were no intervention or cause or an effect analysis. Viral isolates were collected from dead birds and sequenced. The sequences show the genetic diversity of a local gene pool of virus in Houston at a point in time. The samples were taken over a period of 1 year and they're cross-sectional. Also, it was retrospective in that investigators looked at viral sequences from archive samples since 1999. This functions somewhat as a prospective study as we can recreate the evolution of the virus from the founder sequence. In some sense, the founder sequence is the case control, and the cohort sequences are compared to the baseline sequence to ascertain the evolution, meaning the number of mutations of the virus. A mutation presumes change from some accepted sequence. I would say the study is also ecologic in that it reveals the evolution of West Nile virus in the Houston region, where migratory birds are plentiful. The study also had a synthetic aspect in that it relied on existing data, archived virus sequences, for modeling and testing hypotheses, and the model mutation rates. What did the study find? There is ample diversity in the West Nile virus. Some of the original strains have died out. Other strains have been introduced into the population around Houston from migratory birds in other parts of the Western Hemisphere. Continued surveillance is needed. Detailed phylogenetic analysis indicated 3 independent clusters, or clades, which indicated sequence homology with viruses from other regions in the U.S. Okay? To quote from the authors, continued investigation over time is critical to understanding the spread of the virus, viral and host factors contributing to virulence and mosquito competence, and the possible future of vaccines or drug candidates. Thank you.